So I'm super excited today to introduce Dr. Donna L, who will be giving today's seminar. But before I do, I just want to acknowledge that we live, work, and play and participate in a community of the unceded, ceded, and traditional territories of the 203 First Nations, along with 38 Métis chartered communities, each of which possess their own unique traditions and history here on this land that we now refer to as British Columbia. And I recognize that not all of you are in BC or in Canada, but I urge you all to understand and acknowledge the Indigenous communities of lands on which you reside. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the United Nations Declaration on the Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all of our work, we're committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. And today, I'm joining you from Vancouver. I'm on UBC campus right now, which is part of the unceded homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. And this series is part of the Women's Health Cluster, and we have over actually 150 members now. Worldwide, I think we have 17 countries now represented, which is amazing. We hold a number of events. One thing that we haven't announced publicly yet is that we'll be holding another Women's Brain Health Conference in probably November on hormonal contraceptives and the effects on brain health. We have a number of speakers lined up, so look out for that. That's great. We also have currently right now some trainee research awards. So please do, anybody is able to apply for those funds. And it's just, you have to become a member. That's the way we hook you in. You have to become a member before you can apply. But any trainees that are going to be presenting or just attending a conference, please do apply for that. And we want to acknowledge all the people that we give us support because we couldn't do it without them. And again, please do become a member. And with that, I'm going to stop sharing my screen. So while I introduce Dr. Donna L, I will read out not all of her bio because it's quite long and extensive. I'll just read out part of it. So Dr. L joined the Department of Biology at Syracuse University as an associate professor in August 2012. Her research program tests how changes in hormones, metabolism, and cell signaling modulate brain and cognitive function across a lifespan. She and her team have found that hormones such as estrogens can both enhance and impair cognitive function depending on the problem being solved. And I love it because she sees both things. And we see some of these kind of different things as well, which I think is great. And one goal of her research is to understand the role of astrocytes in brain aging under healthy and pathological conditions such as Alzheimer's and Parkinson's disease, and how menopause may mark an important point in the life history of a woman. So absolutely. So excited to hear her speak today. She's published many papers and has funding from NIH, NSF, and private industry. And together, Dr. Korol's research program has important implications for maintaining brain function across aging and health and disease. I know myself, I'm very looking forward to listening to her speak. I am as you know, I like to talk about fun fact, and Dr. Korol has many. So let's see, which one am I going to tell you? I think I'll tell you about that she's an impulsive, if not compulsive baker, she says, and a caretaker to 2 sourdough colonies. So with that, I will welcome Dr. Korol. So thank you so much.
Thank you. And let me share my screen. I think this is it. Yeah. So can everyone see that okay?
Yeah.
Okay, great.
Yep. Perfect.
All right. Well, thank you so much for the nice introduction and for inviting me to speak for the seminar series. The Women's Health Research Cluster is such an important network of internationally recognized people with these critical missions to promote health equity for women and girls worldwide and so many other things. I'm really thrilled to be a part of it and to share my thoughts with you today. My only regret is that I could not be there in person, so someday perhaps. And thank you for the fun fact. I actually, during COVID, started 4 different sourdough formulas. My husband accused me of being not just a compulsive baker, but a compulsive chemist and experimental design enthusiast. So I'm now down to 2. One died and the other I couldn't deal with. So anyway. In addition to baking, I'm interested in the neural mechanisms of learning and memory, as Lisa mentioned, and how they change across the lifespan in health and disease, with a particular focus on how menopause may actually play a very important point in the life course of females and sort of change that trajectory of aging. So as you might be able to tell from the title, I'm going to discuss work that tests how estrogens and the loss of estrogens with menopause can regulate metabolism, which in turn can modulate different types of problem solving. Okay, oopsie. So in addition to the basic science of estrogen's actions, which of course is interesting, there are many important reasons to study the effects of estrogens on brain function and cognition, as noted here by these various conditions in context that might interfere with brain function, both exogenous exposures to estrogens, that might include environmental estrogens, treatments for cancers, hormone-based contraception, but also endogenous factors such as age, hormone status, and sex as variables that would contribute to health and disease. So my lab has actually been developing models of menopause to understand healthy aging in females and how changes in hormone status can increase risk for certain diseases, especially Alzheimer's disease and Parkinson's. So menopause has been connected to metabolism in Alzheimer's and metabolism and cognitive aging. And what's been driving our work is this idea that menopause may actually alter metabolism, which in turn then changes the kind of trajectory, as I mentioned, of cognitive aging through the loss of estrogens. Now, of course, it's also true that changes in function, changes in activity, brain activity, mental activity, even physical activity, might actually in turn change metabolism, both peripheral metabolism and brain metabolism. So teasing apart these two different kind of arrows, I think, is one of our goals, and maybe have important implications for developing targets to help combat cognitive aging or maintain health as women age. Okay, so we think this for many reasons, and for example, women who go through transitional, chemical, or surgical menopause earlier in life are at greater risk for cognitive aging and age-related diseases such as Alzheimer's disease. Taking estrogens around the perimenopausal period protects against age-related cognitive decline, and so the loss of estrogens may actually contribute to risk. Let me just do something. Hold on a second. Something is blocking my screen. I'm very sorry. Okay. I couldn't see what was under there. Okay. The loss of estrogens at menopause increases risk of metabolic disorders, and hypometabolism decreases brain metabolism. And this is supported by findings by Roberta Diaz-Britton and Lisa Mosconi and their colleagues, showing that if you look at PET images of glucose consumption in pre versus postmenopausal women, you can actually see that there's a largely reduced glucose metabolism in the postmenopausal woman. Now, they've also looked at longitudinally as women transition from premenopausal to perimenopausal to postmenopausal states and see the same thing. So within an individual, you can actually see the change in brain metabolism. Now, one thing I want to draw your attention to that's often overlooked is that if you look at this little center area right here, this is a region that tends to be relatively spared in terms of the glucose metabolism as women transition. And lo and behold, this area is the basal ganglia. And as I talk through my findings, hopefully you'll understand why I'm interested in this idea that maybe the basal ganglia function may actually stay intact with the loss of hormones. And finally, we know that Alzheimer's disease and cognitive aging, but especially Alzheimer's disease, is associated with hypometabolism and glucose dysregulation in specific brain regions. And actually, these may be key features of even early stages of Alzheimer's disease that can be attenuated by estrogens. Thus, it's likely that estrogens actually protect neural health through the balance of energy metabolism and provision of energy substrates, and that menopause may actually put women at risk for this cognitive aging. So this is the work I'll describe today that hopefully supports this possibility. So if there are only a couple of items that you take away with, I hope that these are included in what the take-home is. So first is that estrogens act to bias learning strategy by enhancing place learning, and I'll be telling you that's hippocampus sensitive place learning, and impairing striatum or basal ganglia sensitive response learning by modulating the relative contributions of different neural systems. The second take-home message is that we think it's doing so by regulating the metabolic state of each neural system of the hippocampus and the striatum through the regulation of astrocytic function. And so we're going to be focusing on those other cells in the brain, not the neurons, but the astrocytes, even though I do care about neurons too, of course. So the general approach that I'm going to talk about today is outlined here. And although we have used naturally cycling rats, and actually, so in ovarian intact rats and reproductively senescent rats, what I'm going to tell you about is our use of a surgical menopause model to model menopause in women. Now, so what we do is we remove the ovaries and we replace with various regimens of hormones. But again, today I'm going to tell you about mostly the work that we've done in young adult animals. However, we have looked at middle age, which might be the important age, of course, if we're looking at menopause, and old rats. I hope I'll have time to tell you some of the work that we've been doing in aging. We ovariectomize 3 weeks prior to taking our measures. We administer various estrogens, but primarily estradiol benzoate, which is a conjugated form of estrogen. Whoopsie, I'm sorry about that. And the oil vehicle, which is a vehicle control, for 48 and 24 hours prior to the measures that we've been taking. In a subset of animals, we perform cranial surgeries to implant guide cannulae and other types of hardware into the brain. And we do that 1 week after ovariectomy. And then we take the measures in a single day, 3 weeks after the animals have had their ovaries removed. And these include in vivo biochemical monitoring, tissue sampling, and of course cognitive testing, because we're interested in learning and memory. Now, our estradiol injections actually across 2 days mimic, as you can see here, the changes seen across an estrous cycle. So they're not to mimic hormone replacement therapy. We have done some chronic exposures, and I'm not going to have time to talk about that today. And this is an example showing what happens when we give high doses of estradiol, which lead to physiologically high levels of estradiol in the blood. And if we give our lower dose, we tend to see the values hovering around 25 picograms per mil, which is perhaps what people think is a little bit more physiological. Okay, so we use this paradigm to show the changes in learning and memory, and I'm going to describe these shifts in abilities. I'm going to hopefully tell you where in the brain we think this is acting, and you can probably guess by now, and how it's mediated, focusing on metabolic substrates as the cellular mechanisms, and then end with some kind of current and future directions. Okay, so I'm going to start with shifts in abilities. Now, when I first started this work, I reflected on this, it's been over 20 years ago. I was looking at synaptic mechanisms of brain aging, and I was struck by how little was done in females looking at brain aging. And this was with Korol Barnes at University of Arizona. I then realized how little was done to understand learning and memory mechanisms in females at any age. And so I started to focus my research on this. Now, at the time, there was a handful of papers, one of which was Lisa Galia's paper, a set of papers showing us that estrogens had actually mixed effects on learning and memory. And in fact, she found some impairments in some tasks, and I'm going to allude to that in a little bit. And at the time, people thought that the field was a mess because there were not a lot of consistent results, even by this handful of papers. And Vicki Lewin was part of that kind of early group as well. So this actually got me started thinking that, first of all, that we really need to look at females as their own paradigm, but that we really need to understand what estrogens are doing, and in young adults, and also as we transition through aging. Now, because of all the work of people like Lisa and Vicki Lewin, and now many others, there are now thousands of papers that actually have shown that estrogens do have very robust effects on cognition. However, one of the problems is that we still find mixed effects that kind of put the field in a little bit of a kind of a tizzy. So whether we see enhancements or impairments actually tends to depend on a variety of factors, as you can see here. The hormone duration, dose, regimen, the age, the parity history, which I know the Ganglia lab has been very interested in, as well as stress status, the type of memory, and the type of task. Now, what's interesting is that, oopsie, sorry about this, I'm just having trouble. Many of these variables actually differentially engage two memory systems in particular, the hippocampus and the striatum. So, to decipher these mixed effects, we decided to focus on the contributions of these two brain structures. So, not necessarily very complicated thinking, but very straightforward thinking. So we started to develop single solution tasks that specifically tap into the function of these two brain regions to try to understand how estrogens can both enhance and impair learning. So in the place task, as you can see here on the right side of the screen, rats are actually trained to go there, to actually go to a place in the room to find a food reward in the maze. So regardless of where they're started, the goal is always in the same position in the room. For our other kind of task, for our response training task that we know taps into the striatum, whereas the place task taps into the hippocampus, rats are trained to find food by turning this way, by making a body turn to find the food. So if they're started here, they would find the food as it turned to the right. If they were started here, they would turn to the right. In this sense, they are disregarding the room cues to follow their body turns. Now, one reason these tasks are so useful for dissecting out the enhancing and impairing effects of estrogens on learning strategies is that they're matched on what we would call non-mnemonic variables like stress and the training regimen, because everything is the same about them except for the strategy used to solve it and what we think is the brain region that's responsible for solving it. So then measuring learning on these tasks can actually serve as a readout for the relative function of these different structures, the hippocampus and the striatum. And in this way, we can use these to also model different disease phenomena, such as those that are happening in Alzheimer's disease versus Parkinson. So we can actually compare some of the neural mechanisms by using these tasks. So we are interested in, of course, understanding these mixed effects. And so our first question is whether estrogens, specifically estradiol benzoate, would modulate strategy use. So we took separate groups of female rats, and they were trained on either place or response versions of these tasks. And what you can see is shown in this slide. And what we found is that estradiol, actually, if you look here first, actually enhanced learning of the place task. As you can see, by trials to reach criterion were significantly lower, meaning that it took them fewer trials, and so they were faster at learning this task, place learning task, under the influence of estrogens. On the other hand, for response learning, the striatum sensitive task, estrogens actually impaired their ability to learn this task, so it slowed down their learning. They took more trials to reach criterion. What's important to note is that actually if you look across the tasks, that rats under low hormone states actually were better at learning response than they were at learning place. So in this sense, we would expect there to be some conditions where people, for example, or individuals with lower hormones might actually be better at some tasks than others. And if you look over here, you can see that this shift in learning ability is also seen in the percent correct across training, which we call their learning curves. So you can see estradiol accelerated place learning, and estradiol actually impaired or at least delayed response learning, as you can see here. So I had a graduate student now it's been about a decade ago, so time really goes fast. But anyway, Samantha, who's an MD PhD student who was really interested in this sort of effect of exposures of exogenous hormones and the health consequences for women. And so in addition to a variety of different estrogenic compounds, she was interested in looking at the estrogenic compound genistein that's extracted from soy, and is also actually found in a lot of foods, unfortunately, including soy milk and soy lecithins that are added, or at least things that are added to foods. And what she asked is, again, whether these kinds of estrogens, which we actually know bind more tightly to the estrogen receptor beta, but whether these kinds of estrogens interfere with learning and memory, mimic estradiol's effects, and so forth. And so she exposed young adults, ovary-actomized animals, to 2 days of oral genistein. And what she found is, lo and behold, very similar results, almost exactly what she found with her estradiol results. And I'm not showing that just for simplicity, but you can see here that genistein actually enhanced place learning, but also impaired response learning. So she found very similar effects to estradiol. So it tends to mimic the effects. Now, again, I had already mentioned this, but I just want to reiterate that with declining estrogens, we'd expect to see improvements in some in cognition in some contexts and impairments in cognition in other contexts. And so we really need to keep that in mind as we start thinking about targets or estrogenic targets for treatments. Now, because most problems we solve in our daily lives really aren't single solution tasks. They're actually much more complicated, where we actually have multiple solutions or multiple strategies to the same goal or to the same problem. So we actually wanted to kind of make this a little bit more maybe appropriate, sort of valid. And so we said, yes, whether estrogens actually bias learning strategy when both place and response strategies are available, when the animal is given a choice. So to do this, we used a task called the dual solution task. Some people call it the ambiguous T-maze task because we don't know what strategy the animal is using during the training. And on this task, it was actually first developed by Ed Tolman in the mid-20th century and been resurrected since by Mark Packard and others, including us. And what it entails is you start, the animals always started from the same arm, and the goal is always in the same arm during training. And so all they have to learn is to go from this position to that position. Now, as you can tell, hopefully from this figure, that during training, the animal could actually use a go there strategy or a place learning strategy where they follow the room cues, or they could actually use a turn this way or response learning strategy. So during training, we don't know which one they're using. And once they reach criterion, though, we give them a little probe test and we ask them essentially to tell us which strategy they're using. And of course, because they can't speak, they can show us with their behavior. And if you start them from the 180 degrees from where we train them during the probe test, and you give a food reward in either arm, you can actually see that if a rat turns right and goes away from the place where they were trained, then they are most likely using a response strategy. However, if a rat goes back to the same place, thereby making a left-hand turn, in this case, a different body turn, then they're most likely using a place strategy. So we did this, and we actually have done this across the estrus cycle, but I'm only going to show you the results from animals that have been treated with our 2 days of estrogens. And what we can see is our prediction from our previous work was that animals with estrogens do better at place strategies. So therefore, maybe they choose place strategies over response strategies. So what we found is that if you look at the estradiol treated rats and you look at percent of the rats using one strategy over the other, you see that the animals with estrogens had a preference for place learning strategies, and it was a significant bias. If you look at the oil-treated animals, it's not like they were 50-50. The oil-treated animals actually had a preference for response learning strategies. So they're actually preferentially using the striatum-sensitive strategy. Now you might argue, I already told you that estrogens actually speed up learning in some cases. So maybe these estrogens are making them learn faster, and so that makes them use place strategies for some reason. So if we actually look at their learning rates, how long it takes them to learn this task, it actually only takes them about 20 trials. It's not very difficult because they have these different strategies at their fingertips or paw tips or whatever. We see that there's no difference in learning rate at all. And so the rats on estradiol learn at the same speed as the rats on oil. So in this sense, we think that what is happening is that the hormonal status actually regulates what is learned, not only how much is learned. And I think this is a really important take-home, that it shifts their learning strategy. Again, not consistent with findings from Lisa's lab and other labs now too. So because estrogens can actually impact this is just to summarize now, I think from a large number of people, excuse me, we think that estrogens can enhance lots of different strategies that seem to tap into the hippocampus, and that estrogens impair lots of different strategies that seem to tap into the striatum. So we think that estradiol actually acts at the hippocampus to improve learning, and acts at the striatum to impair the learning that we're looking at. So of course, if we want to look at where estradiol is actually acting in the brain, we know that when we give peripheral injections, it can actually go anywhere in the body, but it can certainly go anywhere in the brain. So we started to focus our attention on looking directly at estrogen effects in the hippocampus and striatum. Again, pretty simple thinking, not too complicated. And what we did was we actually used two different approaches. And one was to infuse estradiol directly into the hippocampus or the striatum, and these are always separate groups of animals, and then train them on either place learning or response learning tasks. Again, those are separate groups. And so what we found that when the estradiol was infused into the hippocampus, we saw improvements in place learning. When the estradiol was infused into the striatum, we saw impairments in response learning. This mapping onto what we saw with peripheral estradiol injections. Now, when we infused the estradiol into the hippocampus, we saw no effects on response learning. And when we infused into the striatum, we saw no effects on place learning, suggesting that the effects are independently acting independently at these two different structures. So it's not as if this increase in place learning is making them deficient at response learning. This increase in place learning doesn't seem to be making them deficient at response learning. It's as if estradiol needs to act at the striatum in order to see the impairment in response learning. So then the second approach we took was to actually block estrogen receptors with sort of a pan-estrogen receptor blocker, ICI 182780, prior to systemic estradiol injections. So we had the receptors blocked. We gave them systemic estradiol injections to test for the necessity of these receptors in these different brain regions. And we blocked the estrogen receptors in the hippocampus and trained the animals on place learning tasks. We blocked the receptors in the striatum and trained them on response learning tasks. And what we found is that the estrogen receptor blockade abolished both the enhancements in place learning and abolished the impairments in response learning. So what we think is that estradiol is actually enhancing place learning through estrogen receptor activation in the hippocampus and impairing response learning through estrogen receptor activation in the striatum. Now we know that the estrogen receptors are found in both structures. However, they're distributed quite differently in terms of distributed through the different cellular and subcellular components. So we know that estrogen receptors can be found in the nucleus in the hippocampus and in extranuclear sites. But in the striatum, we know that the estrogen receptors are found only extranuclearly, so they may signal very differently. So we next wanted to ask, since we know that estrogen receptors are necessary, we wanted to ask whether the different effects, the bidirectional effects, the enhancements and emplacements, are due to activation through one receptor versus another. And we had some reasons to believe that estrogen receptor beta may actually be what we would call the enhancing receptor, and estrogen receptor alpha would be the impairing receptor. And I can talk during the discussion about why we thought this if there's questions. We also thought that maybe rapid versus slow, short versus long exposures would explain the bidirectional effects. Now, I'm not going to have time to show you the results, but I just want to tell you them. And that is that the opposing effects did not sort by estrogen receptor subtype or by timing. That is, all three of the receptors we tested actually both enhanced place learning and impaired response learning. And both kind of short versus long exposures enhanced place learning and impaired response learning. So this suggested to us that these, at least alone, are not responsible. And we actually have some complicated results suggesting that interactions may be explaining something, but I'm not going to talk about that today. So at the time we were doing this work with the estrogen receptors, we turned to metabolism because not only because of its relationship to estrogens, but also we were starting to look in males. I'm having trouble finding my cursor. There we go. We were starting to look in males at the role of astrocytic metabolism as a key regulator of learning and memory. And so we did focus our attention on astrocytic metabolism as a metabolic substrate. And here you can see a beautiful glycogen molecule. And the reason I'm showing you that is because one of the kind of unique features of astrocytes and functions is that they actually store glycogen. It's the only glycogen storing element, well, the glial cells are the glycogen storing elements, but the astrocytes primarily in the brain. And the neurons don't store glycogen. And as you can see here, this is just showing you a stain where the green, it's a double stain, where the green is the cell stain and the red is the glycogen stain. And on the left you see the cell stain for astrocytes, and where there's yellow, it's overlapping with the glycogen stain. So you can see that the astrocytes store glycogen, and here the cell stain is a neuronal cell stain, and there's no overlap between the glycogen and the neuronal stain. So there's lots of, not just our evidence, but lots of evidence from others too, that glycogen is stored only in the astrocytes. So, okay, so the brain is a very hungry organ, as many of you know. It consumes upwards of 20% of our the body's energy, while only weighing in at about 2 to 5% of our body mass. So it needs to regulate its provision of energy substrates to meet its very high metabolic demands. And also, as many of you know, the brain gets its energy from glucose, from the blood. Now, what happens next is shown here in what I'm calling our working model, which we adapted from Pierre Magistretti, highlighting astrocytes' critical role in brain function. So first of all, glucose can enter the brain and go directly to the neurons for its energy needs. But glucose also flows through a second pathway, and that is that it can go to the astrocyte, taken up by the astrocyte and converted to lactate, and then used for its own energy needs, or it can actually be shuttled out of the astrocyte, kind of provided to the neuron, or it's used as a signaling molecule to actually stimulate the blood vessels to dilate and perhaps deliver more resources to the brain, including glucose and oxygen and other good stuff. Now, glucose can also be stored as glycogen for later use, as I mentioned already, where it's broken down following activation of the astrocytes through beta-2 adrenergic receptor activation. And that can be done through substances like norepinephrine, or noradrenaline, as it's sometimes commonly called. Now, the ketone bodies also, I think, are big players, and Roberta Diaz-Britton has a very interesting growing story about ketones, and their ketones have received a lot of interest because of ketogenic diets and ketogenic treatments and so forth. And we have an interest in this, but we don't really have much of a story, so I'm going to ignore ketones for today. So we hypothesized that estradiol is increasing these metabolic substrates in hippocampus and decreasing the substrates in striatum. And a lot of this is based on the findings at the time that we know that glucose is a key ingredient for learning and memory in males, okay, and also lactate. And so this work was done by a PhD student, Wei Wang, and in collaboration with a star undergraduate, Ella D'Amico, in the lab. And what she first asked was literally whether extracellular fluid of glucose and lactate is affected by estrogens, actually, in the hippocampus and striatum. So she used this in vivo microdialysis method where she inserted a probe into these different structures, and she sampled the extracellular fluid, and then offline she analyzed it for her analytes of interest, including glucose and lactate. She also took whole tissue samples of ketone bodies. She couldn't measure that in her sample just because the levels were so low. And those were done in post-mortem samples. And what she found is shown here. It's summarized here. You can see in the hippocampus, estradiol increases glucose, and these are just millimolar concentrations in her dialysate, and lactate significantly above those from the oil-treated controls. And actually no dramatic change in tissue content of ketone bodies with her estradiol treatments. Now in the striatum, she found that estradiol actually decreased lactate and ketone bodies, pretty remarkably, with no effect on glucose concentrations. So we were pretty excited about this because this is like the first time we saw a kind of a biochemical or any kind of mechanistic measure that actually matched our effects of estradiol on learning and memory. And so this might actually explain our bidirectional effects, where we see the increase in place learning and the decrease in response learning. So our hypothesis was that if estrogens are increasing glucose, then that suggests that the oil-treated animals might actually be somewhat deficient in hippocampal glucose. So that would mean that replacing the glucose to the hippocampus in these oil-treated animals, the estrogen-deprived animals, would actually mimic the effects of estradiol or replace the effects of estradiol. And so what Wei did was she actually took animals and she infused glucose into the hippocampus. Now, because food restriction is going to affect their metabolic state at baseline, and our tasks in the past required us to engage the animals in some food restriction for about a week before we trained them so they'd be motivated to perform the task, we turned to a different task that didn't require any food restriction. And we turned to a task, it's a working memory task on a simple plus-shaped maze that just requires the animals to navigate this maze, and it's called spontaneous alternation. Now, percent alternation is a measure of working memory in this task, and again, we can talk about how or why this is so in the discussion. Please just trust me for now. And so what we found is that the estradiol treatments alone, just the estradiol treatments alone, enhanced memory on this task compared to the oil-treated animals, but also compared to chance. So they were above chance on this task, and the oil-treated animals were not, actually. Now, so what then Wei did was then she, in a separate group of animals that were treated with oil or estradiol, she infused either saline or glucose into the hippocampus at a dose that we know enhances performance in males, 15 minutes prior to working memory testing. And what did she find? Well, what she found was that in the oil-treated animals, the glucose infusions into the hippocampus restored working memory to the levels that we see in estradiol-treated animals. Interestingly, in estradiol-treated animals, we saw no additive effects of glucose. That is, that glucose enhancement we didn't see any further glucose enhancement of working memory beyond what we saw with the estradiol treatments. So this suggested to us that estradiol-depleted rats were glucose deficient, and estradiol-treated rats have adequate glucose supplies. Okay. So we then wanted to know, what is glucose doing and which pathway is it following? Okay, so it could be that this increased glucose is going to the neurons. It could be that this increased glucose is going to the astrocytes, being stored as glycogen, and then when the animals are actually trained on the task or tested in memory, it's converted to lactate, which is then either given to the neurons for its energy needs or signals to the blood vessels to deliver more glucose. And we know that from males, actually, if you block glycogen breakdown, so you block glycogenolysis with drugs, that glucose no longer reverses memory deficits or enhances memory, but lactate does, which suggests that glucose is upstream from glycogen and lactate is downstream from glycogen. So if you block glycogen breakdown, glucose no longer can enhance memory or reverse this deficit that's seen with glycogen breakdown. If glucose were able to reverse it, it would suggest that it would be acting through a separate pathway. But because glucose no longer reverses it, we think that glucose, the sort of additional glucose, is acting through this astrocytic pathway. So our hypothesis was that estradiol-induced glucose is stored as glycogen for later lactate production and then shuttled out of the astrocyte to enhance memory, whether it's through the neuron or the signaling, neuronal energy, or signaling, we don't know. So what Wei did was she actually measured glycogen in either untested animals that remain in a home cage or memory tested animals that were tested for 20 minutes on this working memory test that I described. And so if you look in untested animals, you see that in the hippocampus, estradiol nicely increases or robustly increases glycogen significantly above the oil treated controls. Now, under memory testing, what we see is that glycogen is relatively depleted in the estradiol treated animals, but not much happens in the oil treated animals. And so what we think that this means is that the glycogen depletion in the hippocampus in the estradiol treated animals reflects the engagement during learning and memory, and sort of the use of this extra glycogen to support the memory. So if you recall, these oil treated animals did not do very well on this working memory task. Now, just to show you that in the striatum, we see that estradiol has no effects on glycogen storage. And that doesn't surprise us because the glucose was not different in the striatum of estradiol-treated animals compared to their oil controls. However, you still see depletion actually during after testing. And we think that this actually just reflects the engagement in the task, but not that estradiol actually works through the striatum to enhance performance on this task. Okay, so the striatum is activated, but estradiol has no effects in the striatum in terms of glycogen storage or depletion. Okay, so far I've told you that estradiol increases glucose in the hippocampus, and it's stored as glycogen. And then during memory testing, we see that there's this depletion of glycogen. Now, if our hypothesis holds true, we would expect to see a concomitant change in lactate, where we'd see an increase in extracellular lactate that is actually reflecting the breakdown of glycogen. This loss of glycogen would be producing lactate, and if it's shuttled out of the astrocyte, we would expect to see increases in extracellular lactate. And so what Wei did is she used these wireless biosensors that allow her to take 1-second readouts of lactate while the animals are engaged in this task. I'm not going to go through the technology, but it's a pretty cool method. If anyone's interested, I'm happy to talk about it. And what she found is that is shown here. So if you look at this is actually a readout of lactate concentration in the hippocampus. And you can see, first of all, that estradiol-treated animals have higher baseline lactate than do oil-treated animals, which corroborates her findings with her microdialysis data, where she actually sampled the extracellular fluid. So that was nice to see this, this replication, essentially. And that this increase is maintained throughout testing. So the estradiol-treated animals always have more lactate throughout testing. And this just shows you the quantitative data for the baseline lactate. Now, if you look at this, the testing-induced change that was resulting from estradiol, you see that estradiol actually potentiates this lactate response to testing, where you can see that compared to, now it's all relative to baseline, you can see that the estradiol-treated animals are putting out a lot more lactate relative to their baseline than the oil-treated animals. And this is seen at the beginning of testing, but throughout testing. So it's not just the initial arousal, we don't think, from being placed on the maze. We actually think it has to do with the cognitive energy or the cognitive burden that the performance on this task is producing. And you can see that there's this increase in the testing-induced change with estradiol both early and late in the testing. So right now, so at this point, we think that actually that lactate is a key player. Oh, and I meant to mention back here, let me just mention also that actually if you look at glucose, you do not see that estradiol actually increases glucose, extracellular glucose during training, which also suggests that it's the astrocytic pathway that estradiol is acting through. The second point is that she's a great surgeon, but occasionally she has missed placements. And so in the few cases where her misplacements were in the perihippocampal region, she actually found that there was a subtle increase, a testing-induced increase, but estradiol did not significantly potentiate that. And if anything, the estradiol effect was that there was lower lactate in those cases. So this is we think this is selective to the hippocampus at this point. So this focuses our attention on lactate and also really on the astrocyte as having a starring role for estradiol's effects on cognition. Okay, so if I have a few more minutes, I'd like to tell you about our work that we're doing now. And I think it's particularly important because of our interest in menopause. And how can you study menopause without looking at animals as they get older? So it's our project that we call Aging and Saging, and I always like to make sure people know this is not a typo. It's not sagging, even though that does tend to happen too, and we should embrace that, but that's a whole separate conversation. But that we find that in males, and this is why this becomes important, is that with age comes some improvements in memory, which really goes against kind of the dogma that we see sort of generalized cognitive decline with aging. So I'm going to hopefully have a minute to show you that. But first I want to show you what we've seen in old females. And again, this is the work of Wei Wong, this great graduate student, and one of her undergraduates, Luis Castellan. And what they found is that we took young and old Fisher 344 rats, which is one of the NIA's models, for better or for worse. And I'm not sure they're the best animals to test cognition, and you'll see why in a second. And we treated them the same as we did for all the work that I've just described. So they are ovariectomized for 3 weeks. We gave them estradiol or oil just before testing. Now, interestingly, what we see here is shown for the hippocampus sensitive place learning. We actually see, if you look across all treatments, we don't see a dramatic age difference in place learning in this case. But we do see that estradiol actually enhances place learning in old Fisher 344 rats on our task. We did not see that estradiol enhanced place learning in the young rats, which is actually kind of troubling, and we've been trying to follow this a little bit to understand whether this is a dose issue, a timing issue, or so forth, because a lot of people, and we ourselves, of course, have seen improvements in the young rats, but also in young Fishers. Others have seen this in similar kinds of tasks. In the response learning task, interestingly, we see that there is an impairment, an age-related impairment in response learning. So as animals get older, they get worse at response learning, old females. And that estradiol impaired response learning and continued to impair response learning even in the old animals. So they did not lose their sensitivity to estradiol's effects, either in the place learning task or the response learning task. So for old animals, we actually see the predicted results, actually, place learning enhancements and response learning impairments. But keep in mind that old females do worse at response tasks, maybe not so much worse at the place task. So we then realize that we have this pretty dramatic sex difference, so that these are the data I just showed you collapsed across treatment groups. So here is the place learning, no difference for young and old, and the response learning impairment in old. However, in males, what we see is that old males do very poorly on place learning tasks, but actually do quite well on response learning tasks. And in fact, are enhanced compared to young males at response learning. Now we have some evidence, and I can show you if you ask, we have some evidence that if you turn off the striatum in males, you actually recover the place learning impairment so that you restore their performance so they look just as good as youngs, which suggests that the hippocampus in males is doing fine. It's just being somehow overridden by this more active striatum. But that's an aside. What's interesting is that we see this dramatic interaction between sex and task, where we actually see, in the response test, age and sex, where we actually see that that the females, the old females do worse and the old males do better. And so we're really interested in exploring this response learning difference. Now in male rats, the lactate response to training matches the cognitive deficits in sparing that I've just described. So in males, remember, the old animals are worse at place, hippocampus sensitive place, and the old males are better at striatum sensitive response. And if you look at the lactate response to training now, and we've actually shown that we do believe that this is because of glycogenolysis, breakdown of glycogen and provision of lactate. But you see that the hippocampal lactate response is much blunted in the hippocampus compared to young males, but not in the striatum, in fact is quite spared. Okay, so we think that either lactate isn't the story in the striatum, but it certainly doesn't show an impairment that we see in the hippocampus. So now the lactate response seems to certainly match the cognitive deficits in sparing. In female rats, the results are way too preliminary to even share with you, but we actually have some evidence with our response learning that the training-induced lactate response does not seem to match the rate of response learning in the females, which was also somewhat confusing to us. So we're still trying to sort through those results. But there might be a sex difference in aging and reliance on these metabolic substrates. So hopefully what I've shown you is that estrogens bidirectionally regulate different energy substrates in the hippocampus and striatum, and we think that they actually contribute or maybe mediate the enhancing effects on place learning or spatial memory in general, and the impairing effects on what we would call response learning or stimulus response learning that other people, or stimulus response memory. So for this, in conditions with depleted estrogens, we would expect to see improvements in cognition and in some contexts and impairments in others. And we've also shown that estrogens promote hippocampal astrocytic glycogen storage that may be converted to lactate and then used to encourage more energy substrates to come to the brain, to the hippocampus, or for the neurons to use this lactate as its energy source. And so we think that astrocytic function may actually mediate these hormone effects. And I think the impact of this is really when we start thinking about developing targets for treatments, both that are maybe estrogen receptor blocker treatments, but also for neurometabolic disorders, including Alzheimer's disease, to keep in mind, the astrocyte as an important target for these sorts of treatments. And then, so I'd like to end by acknowledging the current members of the lab, and we no longer have to meet by Zoom, which is very exciting. But I still like this photo because it reminds us of how much better these days are, and it's good to have those reminders. So the current members, Stephen Ajayi, Clementine Harvey, Dalton Myers, Jules Kuki, Taylor Drabick, Monica Chiaffi, Michaela Martin, and Mackenzie Allen, who've all contributed in lots of ways to this work. And then past members, Alicia Prokopenka, who was a postdoc and has a position at Midwestern University in Chicago, which is very exciting. Bob Gardner was a postdoc, Lori Newman, Wei Wong, Luis, Ella, Sam, Claire, Lily, and Stephanie, who've all contributed. I'd also like to acknowledge the Haudenosaunee. So the Onondaga Nation, which is the firekeepers of the Haudenosaunee, are the indigenous peoples on whose, I always like to emphasize, unceded ancestral land Syracuse University now stands. And so again, I'm home, but my house is also on unceded Onondaga lands, and so I'd like to acknowledge that as well. And then collaborators, Paul Gold, who's my kind of collaborator in life and in the lab, so he's my partner and spouse. Sue Shantz, John Katzenellenbogen, and Bill Helfrich at UIUC. My funding, and then a special thanks to our animal care staff, and of course to the rats too that contributed their lives. And thanks to you for your attention.
Thank you so much. That was great. What a great overview of so many different topics. I can't even, that was fantastic. So in our unfortunately still virtual world here, if some of you feel like, doing the little animated claps, that would be fantastic. I see a couple already, which is great. And of course, as always, if you'd like to put something in the chat, you can, or you can unmute yourself if you want to and ask a question. Please feel free to do so, because you know what will happen if you guys don't ask a question. And Jesse Lacase looks like he might have unmuted himself already, so I'm so excited.
Well, let me see if I can. Oh, hi, Jesse.
Hey, Dr. Korol. That was great. Just a quick question for you, and correct me if my way of thinking about this is a little off, but a lot of the time when you hear about lactate and stuff, you really hear about, you work harder, you lift more weights, and you pour out more lactate, and so I'm wondering, you showed the excess lactate in the estradiol conditions, especially during training, you have more extracellular lactate. I'm wondering, are they working harder, or is it something about the cost of using the hippocampus? Why are you seeing more lactate? And does that mean kind of the same analogy in the muscle?
Yeah, no, I like that question. First of all, what's interesting is lactate used to be thought of as a waste product, even for muscles, but now we don't think that way. So it's actually it's been disproved that the lactate is even what causes the cramping. Actually, so lactate has been thought of as an important signaling molecule even in the muscles. But I think you're right. I actually think that the lactate response might reflect the relative engagement of that structure, and so it's getting more beta 2 adrenergic activation, glycogen is being broken down, and therefore there's more lactate being produced. So I do think it's working harder, even though, because we think estradiol improves cognition, it may be engaging the structure more. So whether that's working harder or not is kind of a opinion, I would say, more than empirical. But we do think that it is actually turning on the hippocampus, which then might require the glycogen to be broken down and the lactate to be used to get more resources to come to the hippocampus. And actually, I didn't show the results, but we do have some results where if we use a different kind of spontaneous alternation test, which does not have a delay in it, we actually don't see the same results. It's too easy. And so it actually directly addresses your question, and we don't see the same amount of lactate being produced.
That's interesting. Cool. Thank you so much.
You're welcome.
Does anybody else have any? Oh, Troy, did you? Because I think you came off of mute as well.
Yeah, I was wondering, so about the receptor subtype that would be involved with this for estrogen, because it seems like you're having membrane initiated or rapid effects. So that could be, of course, a number of them, but I was wondering if you had any idea.
Yeah, so do you want, I actually have results for those, but if you'd like to see them, I can show them to you. But we actually find that both, so the effects for the actual metabolic effects, is that what you mean?
Yeah.
No, we haven't done that yet, but that's
Okay.
actually on the docket to look at. And the terms we like to use, we compare PPT to ER beta 041. It's a more selective ER beta agonist that matches ER alpha a little better. So DPN is a little bit of a broader receptor agonist. Yes, it's messier, and that's why we think people found more cognitive effects of DPN at the early stages of this work. Actually, if you don't, well, I can show you the results some other time. But they're in a paper that was published in 2015, I believe. But the dose response function is very different between DPN and ER beta 041.
Oh, that's interesting.
Yeah.
Sorry, Troy, I'm like commenting. Go ahead, Troy.
No, that's fun. That's interesting. I just feel like there's, especially in certain regions where you have overlap of ER alpha and beta, and then you have GPR1, and you could even have the GQ coupled membrane estrogen receptor, that could explain some of the, if you say, positive and negative effects. It depends on the receptor that's expressed, and so I would wager
I'm just going to show you. Yeah, I'm going to just show you the results real quick. Just so if you look at PPT versus DPN, you see that we have a single dose of PPT that enhances place, and a couple doses fairly high, not too high compared to what some people use, but that impair response learning. And then DPN is a little bit broader for the enhancements of place learning. But then if we look at ER beta 041, we see that it's kind of the single dose that overlaps pretty well with the PPT. But again, we see both the enhancement and the impairment. We thought that we would see the enhancement here and the impairment here. And then the GPR one is even different, so. But again, we saw both enhancements and impairments for G1 exposures.
Oh, wow.
But we haven't, there's some good evidence about the estrogen receptors and their distribution to mitochondria and so forth. So, and Wayne Brake's lab, so with Jesse's lab, along with Terry Milner and others, have shown beautiful distributions of these receptors in striatum, in astrocytes and neurons in striatum and so forth. So we use that as our guide, but we haven't been able to run those experiments yet.
Because I would wonder if you had to if you did this in mice, if you could do this in mice and use knockouts, or if you selectively knock down these in the rat in this region or something like that, if you saw differences.
Right, right. Yeah, I think, yeah, the mice are so different in terms of these tasks. They're not
I know that's the
great.
problem. Yeah.
They're great at response, not great at place. These are kind of reactive, but you can do it, and we've used object recognition, similar, a modified version of like what Karen Frick and others have used. And we actually have a striatal version of that, which is pretty cool. So we have used that, but it's a little harder to know when to give the treatments, if it's just before all the testing or the memory testing, because it has a training session and then a testing session. So it's a little more complicated in the design, but we would love to try to know like which receptors it's working through, especially because of looking at like, ER alpha receptor blockade for certain cancer treatments and so forth. So.
Right.
And which receptor on which cells, which I think
Right.
we're also alluding to too. I would love to continue this conversation, but we are over time. So thank you so much for your questions and engagement. And I don't get to ask my 3 questions, one of which I can't understand what I wrote down here anyway, but I will email you. But thank you. Join me again and thanking Dr. Korol for a great talk. Thank you so much.
Thank you. Thank you so much. The pleasure is mine. So.
Oh no. Thank you.
Bye.
Bye, everyone.